the country. You all have heard that the DCGI has approved Biocon's first-in-class anti-CD6 biologic itolizumab for life-threatening COVID-19, and this brings in new hope for the patients. We will hear more from our esteemed panelists today. Let me introduce them to you. We have with us our Executive Chairperson, Kiran Mazumdar-Shaw, who will share her insights into the pandemic, India's relative position, and the current arsenal of drugs that are being used to combat the disease and the effective role that itolizumab is going to play. We are also privileged to have with us on the panel principal investigators of the clinical study, Dr. Suresh Kumar, Dr. Rosemary D'Souza, and Dr. Hemant Deshmukh. They are joined by Dr. Shashank Joshi from the COVID-19 Task Force of Mumbai and Dr. Hemant Thakkar. All of them are constantly leading the fight against corona in the wards of their hospitals. They also led the itolizumab clinical study, which has shown compelling results, enabling us to positively impact patient lives. We also have Dr. Sandeep Atale, who is the Chief Medical Officer of Biocon Biologics, and he will be discussing the results of the clinical study with you in detail. We have seen that there is a little bit of confusion in the market. People are not really able to understand how and why this drug got approval with a very small patient study. But I'm sure once you've heard from Dr. Sandeep Atale, all your doubts will be addressed. We also have several other esteemed members of the medical fraternity here today with us to share their experience. As we move forward, you will hear from them. And I would also like to extend a very warm welcome to a varied audience comprising of pharmacists, medical professionals, and our friends from the media world. So without further ado, I would now like to request our chairperson, Dr. Kiran Mazumdar-Shaw, to open the event with her address. Over to you, ma'am.
Thank you, Seema. I would like to start by saying thank you for joining this press conference. And I would like to basically go through, give you some insights and optics into what this drug is all about, why we have developed the drug, and why we are basically, excited about the data. So let me start by saying that the coronavirus has ravaged the world with this pandemic. And as you know, it has plunged us into a humanitarian crisis and an economic crisis. If you remember, in the first few months of this year, when the pandemic started becoming known, the understanding of the disease was that it was a nasty flu. Of course, there was an understanding that coronaviruses are nastier than influenza viruses, but the exact mechanism of action started only becoming known towards the end of March. This was when you saw a huge mortality rate both in Italy and in the US. And it was at this time that there was a huge effort to understand what was happening and why we were facing so many deaths. Now, as things stand today, there are still very alarming numbers that we are seeing around the world. The world is approaching 13 million cases of COVID-19, and it has crossed 500,000 deaths in the world. When you look at India, we are inching towards 1 million cases, and we have crossed 22,000 deaths as of today. When you look at the US has crossed 3 million cases, and it has, it is inching towards 150,000 deaths. Now, when you look at the death rate per population, India, of course, is still at 1.64, which is relatively lower than most parts of the world. In fact, if you look at the same ratio in the US, it is very alarming. They have 40.99 cases of deaths per 100,000 population. And this is why the world is so alarmed and in such a state of panic, crisis, and not knowing what to do. We need, until the vaccine comes, we certainly need life-saving drugs. And I think what we are doing across the world is to see how we can either repurpose drugs or develop new drugs to treat this pandemic. Now, let me also say that even if we get a vaccine by the end of the year or early next year, there is no guarantee that there will not be reinfection. There is no guarantee that it will work the way we expect it to work. So we must be in a state of preparedness. I would like to start by saying that when we started on this journey of developing and repurposing itolizumab for COVID-19, it was based on a very strong hypothesis that was related to the cytokine release storm, or the cytokine release syndrome, as it is often called in medical terms. This was identified as the leading cause of deaths by the end of March. We looked at this drug itolizumab, which had been developed for its immunomodulatory mechanism of action, which downregulates cytokines, and we know that it is very effective in this cytokine release syndrome. This drug basically was developed in India for psoriasis, because psoriasis is an autoimmune condition that sees a large release of cytokines, and the ability of itolizumab to downregulate cytokines made it compelling to develop it for psoriasis. Of course, we also know that itolizumab works in a host of immune mediated diseases. But instead of developing it in India, which does not appreciate this kind of path-breaking innovation and innovative mechanisms, we felt it was going to be better understood in other parts of the world. And we therefore then licensed it to a US company, which started developing new indications for this drug, again using the same hypothesis of cytokine regulation modulating. And they have started studies in graft versus host disease, which is about the rejection that transplant patients face when they accept a donor organ. And they have also started looking at immune modulation in acute asthma and lupus nephritis. So when all this was ongoing, when COVID happened, we said this actually gives us a very strong case to try it for COVID, because we do believe that this unique mechanism of action that it has can actually help us to deal with this storm that we are seeing that is killing patients. Of course, I must make it very clear to everyone, please don't panic. 80% of COVID-19 patients have very strong immunity and they get away with mild symptoms, and really they don't need this kind of intervention. 20% of patients are hospitalized, and half of these are moderate to severe ARDS patients. These are the patients who come with oxygen deficiency or hypoxia, and they start displaying signs of cytokine release. And then 3 to 5% of the patients become very sick, where they have very high levels of cytokine release syndrome, and they are unable to cope with the kind of excessive cytokines that are released in the body, and many of them then succumb. So with this background, I wanted to inform you that this is the rationale with which we then said, look, I think itolizumab has a very good chance of being effective in these kind of patients. Moreover, I must also tell you that we had 7 years of very good safety data on itolizumab, and we had started understanding the mechanism of action of itolizumab much deeper and much better. And it is with a very strong scientific rationale that we went into this trial. Now, when you look at what is happening, the acute respiratory distress syndrome, what we are seeing is that this virus is actually hyperactivating the immune system. And it is the hyperactivation of the T cells in our immune system that is leading to a massive release of cytokines. And when you have this massive release of cytokines, you actually start seeing organ damage, lung damage, kidney damage, other organ, deterioration, which finally leads to death if you don't intervene. So it was very obvious that we had to start addressing these cytokines and to see how we can basically get rid of them and stop them from replicating in such large numbers. And therefore we said that this is a drug that is anyway down-regulating cytokines. Can we basically make a difference? As a scientist entrepreneur, I felt we must not lose this opportunity to try such an important drug in saving lives. That is the objective with which we went into this trial. Now, I also want to say that itolizumab is a breakthrough first-in-class novel biologic therapy for this particular indication. And I can tell you that when we approached the DCGI with this hypothesis and with this rationale, we had a very deep scientific discussion with the DCGI, and they also felt that it is worth trying, because at that time we did not have many options and we felt that this was worth trying. Itolizumab is an anti-CD6 humanized IgG1 monoclonal antibody. And the beauty of this drug is it is working upstream, which means it actually starts regulating the immune system, rebooting the immune system, and it starts down-regulating the immune system, saying, do not make such pro-inflammatory cytokines. Start slowing down. And by doing that, the body then recovers to fight the virus and recover completely. By effectively controlling, therefore, hyperactivation of the immune system, it prevents morbidity and mortality related to this cytokine storm. Now, when you look at this particular slide, you can see that the way you have to look at administering this particular drug is to see what happens in the disease continuum. So the first phase, or the stage 1, is called the viral response phase. So your body first starts seeing the virus, and the body starts responding to the virus by releasing antibodies. But by this time, the virus has a very strange effect on your immune system. It starts telling your immune system that you've got to be much more active than what you are right now. And that's when you start seeing this huge release of cytokines. And the T cells also are activated. And so what this then leads to is the ARDS, hypoxia, inflammation. And so what you're seeing in stage 2 is the pulmonary stage, where your lungs passages start becoming inflamed. And you're beginning to see the hypoxia or the breathlessness or the low intake of oxygen. By stage 3, when there is hyperinflammation, you will start getting a much worse set of symptoms. And this is both at this stage 2 and stage 3 that you start using drugs like itolizumab. Now, the breathlessness or the low intake of oxygen. By stage 3, when there is hyperinflammation, you will start getting a much worse. Sorry. So when you start looking at the various drugs that are being used today to deal with the COVID-19, you know that you're familiar with anti-malarial drug hydroxychloroquine. This is being used at an early stage, even as a prophylactic, because of the fact that it works on the ACE2 receptor through which the virus enters your cells. And because hydroxychloroquine inhibits that, it is felt that it could have some action. So this is at the early stage. Remdesivir, as you know, is an antiviral drug, and it is also being used in the early stage of the drug, hoping that it can stop the virus. That is the way it is being used, to interrupt the viral multiplication and to see whether it can stop the virus from really attacking, but it's used in the early stage. Then there is a drug called tocilizumab, which is being used off-label, again to start dealing with the cytokine release storm, because and IL-6 or interleukin-6 is a very dominant cytokine in the cytokine release syndrome, which this particular antibody tries to stop after it is formed. So as the cytokine is being released, this drug tries to mop it up, tries to block it. And then you have another antiviral called favipiravir, which is also being, which has also received emergency approval in India for mild to moderate cases. And then we have convalescent plasma therapy, which is again using the natural antibodies or neutralizing antibodies that have been already formed in people who have recovered from COVID-19. And these are then used to fight the disease in the very serious, or even throughout the continuum, you can use this particular therapy to see whether you can actually get rid of the virus. Now, when you look at how do we look at itolizumab and when do we use it, I would just like to present this slide which tells you that these are on the top, you have got the number of days, and then at the bottom, you're seeing what are the kind of drugs you should be using at which stage. So when you look at the first stage of mild symptoms, at that stage, actually, you can use favipiravir or HCQ. The moment you get into hospital, the first thing that people start looking at is remdesivir, because it's an intravenously given antiviral, and prednisolone, which is a steroid which is used to reduce the inflammation. That's what is commonly done. In addition to that, because we know that this cytokine storm also leads to blood clotting and blood coagulation, low molecular weight heparin is also given at this stage. You can also start giving convalescent plasma if you wish. But when nothing of that works, and when the patients start getting into the cytokine storm, this is the time to start giving products like itolizumab and tocilizumab. And then, you know, when people are presenting themselves very late in the disease, when they have to be put on the ventilator, then you can use both biologics and dexamethasone, which is a stronger steroid. So this is the way the doctors are now treating patients, and patients are actually doing much better than before. So you are likely to see much lower death levels. And I'm hoping that, you know, drugs like itolizumab will reduce it even further. So we should aim to get the mortality rates below 1%. That should be our objective in dealing with this pandemic. Now, I just want to tell you what is the difference between tocilizumab and itolizumab. Many people have asked me, is itolizumab a biosimilar? And the answer is no. It is an original innovative drug. This is a drug that we licensed from Cuba 20 years ago. And we developed it in India. We actually did a lot of research in India. We actually did clinical trials in India, and it is a made in India, innovated in India drug, which we have licensed to a US company who is also doing trials with the drug. And we are very proud that this is actually an original first-in-class drug. There is no other company in the whole world that has an anti-CD6 antibody. So this is a unique drug. There are many companies who have an anti-IL-6 drug, but we are the only company in the world with our Cuban partners who have an anti-CD6 drug. The mechanism of action, I don't want to make it very technical, but what I do want to say is that this works upstream, which means that CD6, which is on T cells, which I remember I told you that T cells are the ones that actually get triggered off and they fire off and make them produce cytokines. It actually works on the T cell activation right upstream, and it starts down regulating IL-6, IL-2, TNF alpha, and a lot of other interleukins like IL-17, etc. So what the beauty of this drug is, it works upstream. It actually gets hold of the T cells who are actually making all these cytokines happen to say, no, do not make those cytokines. And it starts down regulating it. Tocilizumab on the other side is actually binding to the IL-6 receptor. And so it works downstream, which means that it doesn't prevent the formation, but it deals with it when it is formed. So it is not able to deal with too much of IL-6 when it is being formed, because you have to make sure that it can block all the IL-6 that is being formed. Whereas CD6 prevents the formation of IL-6 altogether. That's the beauty of this drug. So tocilizumab has its own role to play, and itolizumab has its own role to play. And what I want you to understand is that biologics are very specific. The immune system is very complex. And what we found is that this itolizumab is ideally suited in a COVID-like situation. So I just wanted to give you that particular clarification and scientific insights into what this drug is. Because very often people think that it is a biosimilar, which it is not. Now, I wanted to again tell you that this has a drug which has been used over 7 years in India with very high safety profile. And it has been approved in 2013. Over 500 patients have used this. And so therefore, we are very confident that this is a drug that can be used very safely. Next. Now, what I also want to tell you is that as we started our trial, we also learned that our Cuban partners from whom we had licensed this drug 20 years ago also started using itolizumab in Cuba. And what was interesting was that they were also reporting very, very interesting data from this trial. They actually reported that 79 COVID patients who were treated with itolizumab, and in fact, they used it in severely infected patients. We did not use it in people on ventilators. We used it just before the ventilator on non-invasive ventilation. They used it on invasive ventilators. And even there, they found a huge improvement. And many patients were rescued in that trial. So I think this also gave us the confidence that we are on the right track. Then we started sharing data with them, saying that, look, this is what we are doing. They started sharing data with us. They have also come up with very good publications in recent days. So I think that this is a very, very good decision we took to come up with this drug and to use it for COVID-19. It is going to save lives. Now, what we have now done is we then discussed it with the DCGI, and we decided, because it's an approved drug and because there's an emergency-like situation, and in an emergency-like situation, you do small pivotal studies, because if it works, it works. If it doesn't work, well, you tried at least. So that's the way you actually do a lot of these studies. So in this emergency-like situation, we said, okay, we must have a randomized, multicentric study where there should be a small arm which is given standard of care, and the bigger arm should be given the drug, so that if it works, you should actually see the signals that it is working, so that you can quickly make it available for use of patients who can benefit from it. If it was not an approved drug, you would have had to do a much longer study, because you have to first show safety, then you show efficacy. But here, since the safety was already known, we decided that we have to do an efficacy study, which is how we did it. And what was important is that we had also upfront said what should be the primary endpoints. And the good outcome of this trial was that both the primary endpoints and the secondary endpoints. So the primary endpoint for me was that we must stop deaths. Can we save lives? Can we rescue people from death? That was our first endpoint. And the secondary endpoint was to see that this drug is working by reducing the cytokine storm. So we have to measure all the markers of cytokines. And in both these, I'm very pleased to say that we met both these endpoints. This study details will, of course, be taken into much greater detail by Sandeep Atle, our CMO. But all I would like to say is that in the patients, 20 out of 20 patients who were on itolizumab recovered, whereas 3 of the patients on the control arm died, unfortunately. So it basically gave a very, very strong, statistically significant data that then allowed the regulators to give us the restricted emergency approval. Okay, which is how emergency, clinical trials are conducted and approved. Now, so as I mentioned, what is itolizumab? It is a first-in-class humanized recombinant anti-CD6 monoclonal antibody. It is available as a 25 mg per 5 ml solution in a vial, and it has to be, administered in a hospital setting. It is right now indicated for moderate to severe ARDS due to COVID-19. It has a unique mechanism of action to effectively control hyperactivation of the immune system by reducing cytokines. And of course, in terms of safety, we have a 7-year proven track record of safety in our psoriasis data. So I would like to say that we are committed to saving patient lives with Alzumab. The cost of the therapy roughly is 32,000 because each vial costs 7,950 rupees MRP. You need 4 vials per patient. Most of the patients need 4 vials. Some patients need a second dose, which requires an additional 2 vials. So 6 vials is what a patient might need if they need a second dose, but I would say most of the patients need 4 vials. What I also want to tell you is that apart from the 20 patients who recovered in this particular trial, many doctors and investigators in many parts of the country decided to use Alzumab, which is available in the market, just like they're using tocilizumab, to try and see if they can save more patients. And I can tell you, as of date, over 150 patients have been rescued with itolizumab or Alzumab in Maharashtra, Gujarat, and Delhi. So this is something which I want you to know, that whilst the 20 patients were done as a controlled trial, over 150 patients in many parts of the country have also benefited from this drug, because you need hope. You need drugs that can save lives. And so whilst tocilizumab has also been used, itolizumab was also used. That should give you also some indications as to how well this drug is being used in many parts of the country. In the remote cities also, somebody told me that, are you only using it in the metros? But the answer is no. We are also using it in many parts. In Maharashtra, for instance, one of the biggest users is in Sholapur, in Nagpur, Nasik, many other parts of Maharashtra. In Baroda also, in Gujarat, they've been using it, and also in Delhi. So I just want to give you an understanding that this is a drug that has shown very convincing and compelling efficacy in a much larger number of patients than what has just been described in the trial. So with that, I would like to hand it over to my colleague, Dr. Sandeep Atle, who will give you a much clearer, and deeper and detailed, introduction into the trial. Thank you.
Thank you, Kiran. Good afternoon, all. This is Dr. Sandeep Atle. I'm the Chief Medical Officer at Biocon Biologics. And today I'll take you through the COVID-19 study results. Could you move to the next slide, please? Yeah, so as we have discussed earlier, Alzumab, which was an approved drug in India, we went for a label expansion to the COVID-19 complications indication. And early in May, we went to the DCGI to discuss our protocol. And that was a multi-centric, open-label, 2-arm randomized pivotal trial to study the efficacy and safety of itolizumab in COVID-19 complications. This study was done in moderate to severe ARDS patients. The trial centers, we chose 4 trial centers for this study. There were 2 centers in Mumbai, which was KEM Hospital and Nair Hospital. And then there are 2 centers in Delhi, which was LNJP Hospital and AIIMS. So Kiran has walked you through the mechanism of action, so I would not spend much more time here. But I think just to recap what this drug does is the antigen presenting cell, which is what you see in the center, is basically the one that traps the virus, and it presents the antigen from the virus to our naive T cells in the body. So focus on the left side of the screen, where typically these naive T cells, they get activated. And how do they get activated? It is the binding between the ALCAM chain and the CD6 chain. And when this binding takes place, the naive T cells get activated. And in some patients where the immune system becomes dysfunctional, then the further cascade of events takes place, which releases a huge amount of cytokines, and therefore it causes cytokine storm. And that is where itolizumab blocks. Coming to the study design overview, the eligible patients were divided into, randomized into arm A and B. There are 20 patients on arm A who received itolizumab and the best supportive care that was available in the hospitals. And arm B were 10 patients who received the best supportive care. Overall, this treatment period was of up to 4 doses. And the end of the protocol, from screening to the end of the protocol time was about 4 weeks. Next slide, please. Coming to the enrollment and demography, we randomized a total of 32 patients in the trial. Arm A were 22 patients were randomized in Arm A, and Arm B, 10 patients were randomized. There were 2 patients who were randomized but discontinued prior to dosing, and that's why they are replaced. At the end of the study, all patients had completed the study, and as mentioned earlier, there were 3 deaths in Arm B. Just to remind, in the trial, we gave the first dose, which was 1.6 milligrams per kilogram given as an IV infusion. And the subsequent doses, which were weekly doses, were at 0.8 milligram per kg dose, given as also as an infusion. Coming to baseline demography, if you look at the mean and the median age in both the arms, it was 50 years and 48 years of age. So they were very well balanced. Particularly to focus, there were 15 patients and 8 patients in both the arms who were less than 60 years of age. But there were also elderly patients who were greater than 60 years of age. There were 5 and 2 in both the arms. In terms of the gender, I think it was predominantly a male participation in the trial, as we expect. And in terms of comorbidities, there were also patients who had comorbidities such as diabetes, hypertension, and hypothyroidism. Now, coming to the efficacy, let me walk you through the 1-month mortality rate, which was the primary endpoint in this study. As we indicated earlier, on the left side, you can see in Arm A, out of the 20 patients, all 20 patients recovered by day 30. And in Arm B, there were 7 patients who recovered, and unfortunately, there were 3 deaths in Arm B. So we have done a statistical analysis on the mortality rate. And as you can see on the right side, we got a very highly significant statistical p-value. Typically, a p-value less than 0.05 is considered very statistically significant. So we received statistically significant results, and the primary endpoint was met. Now, there are some other efficacy parameters that are of interest, and these were stable PaO2 without increasing FiO2. What it means is if the patient can actually maintain their oxygenation without increasing the oxygen flow. And I think basically what we have shown here is if you look at day 7, day 14, day 21, and day 30, and you see the proportion of patients on both the arms, you can see on day 7, it was 90% versus 60%. On day 14, it was 95 to 70%. And day 21 onwards, there was 100% improvement in PaO2 and 70% in arm B. So I think running through the statistics, again, the p-value was highly significant day 21 onwards. And therefore, and again to mention that there were 3 deaths that happened in arm B on day 4, 5, and 12. Another efficacy parameter that is of interest is if the patients had stable or improved SpO2 without increasing the FiO2. Again, this means the oxygen saturation, is it improving without giving more oxygen supplied to them? So again, in this parameter, just like the previous slide, you see the proportion of patients in both the arms. On day 7, we had 90 versus 60. On day 14, we had 95 versus 70%. On day 21 and 30, we had 100 versus 70. Again, when we did the statistics on this, it was highly significant. Another efficacy parameter that is of interest is how many patients actually got weaned off oxygen and how many went on the invasive mechanical ventilator. And in this case, you can see that at baseline, there were about 5 patients who were on non-invasive ventilator on arm A. Arm B had 4 patients. And on day 7, 14, 21, and 30, you can see that 5, 15, 19, and 20 patients progressively came off oxygen on arm A, whereas on arm B, there were 2 patients out of those 4 in the beginning at baseline who actually worsened and went on a ventilator. And eventually those patients succumbed. So there were 3 deaths at the end of week 2, and rest of the 7 patients got weaned off. Again, this is a parameter for those who are interested in this PF ratio. We can clearly see the differentiation between the blue line is the arm A, which is on itolizumab. Arm B was the red line, and you can clearly see that the separation occurs day 14 onwards very clearly. So there was mean change from baseline was 190 in arm A, and it was much lesser in arm B. Coming to the IL-6 levels, I mean, this is a key marker of CRS, and what we wanted to do is to measure these markers to correlate whether these markers are coming down post-dosing. And what we have seen here, as you can see, these are the mean values of IL-6. In arm A, it came down from 178 to 45 post-dose. At the same time, in arm B, it went up from 163 to 211. So there was a sharp decline seen in the mean IL-6 levels in arm A compared to arm B. Almost 4-fold decline was seen in arm A, and there was about 30% increase in arm B. Along with IL-6, another marker which is a key marker for inflammation is TNF-alpha. And just like the previous slide, post-dose, you can see that in arm A, it came down from 45 to 10 post-dose. And at the same time, in arm B, it actually rose from 12 to 39. Another marker of interest for most physicians is the ferritin levels, serum ferritin levels. And they indicate the RBC damage and worsening of disease. So what we saw clearly in A was this is mean change from baseline. So your baseline is now considered as 0. And you can clearly see that by day 7, day 14, day 21, and day 30, how these values came down up to minus 939, it came by day 30. Whereas on arm B, you can clearly see that these values came down a bit, but I think not to the same magnitude as we saw with the treatment arm. Another marker of interest that is commonly done nowadays is D-dimer, and D-dimer levels indicate coagulopathy. That means if there is clotting going on in the body. And I think what we see here is mean change from baseline based on arm A. Basically, you're seeing that there is a consistent decline in arm A over day 7, 14, 21. Whereas if you see on the arm B, you can clearly see that these levels were actually rising post over the time, and eventually never came down from baseline. Another marker which is of interest is LDH, and LDH indicates acute inflammation and worsening of disease. This is where the organ damage starts happening. And I think on the left side, you can see in arm A, again, change from baseline. From 0, it came down over time. On day 7, 14, 21, it gradually came down consistently, whereas these values were little off and all over the place for the arm B. So I think what it does signify is there was a consistent suppression and lowering of LDH values on arm A. One of the things that happens in COVID-19 patients is because of the viremia, there is a decrease in your lymphocytes. And absolute lymphocyte count is one parameter that we check. And because of the mechanism of action of itolizumab, we wanted to see if there is a further reduction in lymphocytes because of its mechanism. But as you can see on arm A and B, there was a slight dip in the lymphocyte count on arm A by day 7 from 0 to minus 44. But again, this was short-lived. It was for 2 or 3 days. And I think by day 7, it started climbing up again. And it closely resembles the control arm. So I think what we gained out of this is the suppression of lymphocytes was not an issue with this product, and the patients were safe. Coming to treatment emergent serious adverse events, what you see on the top is there were 2 serious adverse events that happened on arm A, and the arm B, as I indicated, there were 3 deaths. Particularly in arm A, there was 1 case of uncontrolled hypothyroidism, and that was deemed not related to the study drug. The patient completely recovered. There were high TSH values to begin with. But I think the key point here is that this patient was on a high flow of oxygen before, and he is completely off oxygen and is. On the other hand, I think there was another one, which was an infusion reaction that happened in one of the patients, who was a 51-year-old male. And this was a related event with the study drug. However, this patient has recovered. I think what we learned from this adverse event was that earlier this product was administered over 2 hours. And whenever the rate of infusion was increased, that is where this infusion reaction was seen. And learning from this event, we have now actually expanded this infusion to almost 6 hours. And by doing that, we have not seen these kind of reactions in any further patients. And just recapping, there were 3 deaths again in arm B. They died with ARDS or type 1 respiratory failure. And these happened on day 4, 5, and 12. Let us focus on the treatment emergent adverse events other than the SAEs we saw on the previous slide. So there are 2 events I would like you to focus on, which are of relevance. I think the first one is infusion reactions. We saw 4 in arm A that were related to the study drug. As I indicated in my previous slide, these were manageable, and then we have changed the protocol on how the drug is administered, and now these are much reduced when you slow down the infusion. In terms of infections, we haven't seen anything. In these kind of immunomodulatory drugs, this is something we look for, but we haven't seen, and I think it is consistent with our safety from the past. In terms of transient lymphopenia, which I mentioned in the ALC slide, there were 11 cases where we saw that there was transient lymphopenia, which lasted for 2 to 3 days, and it recovered very well in all the patients. So this was considered related. But other than this, I think the drug was very well tolerated with all the patients, and there were no surprising new things that came up in this particular trial. So I would conclude the presentation by taking you to the few main key points. The first one being the primary endpoint of mortality was statistically and highly significant, and it was in favor of the itolizumab arm. Other key endpoints of lung function, such as improvement in PaO2 and oxygen saturation, were statistically significant in favor of the itolizumab arm. The key inflammatory markers, such as IL-6, TNF-alpha, which support the mechanism of action and support that there is suppression based on itolizumab, are significantly reduced after dosing, and therefore they are preventing the hyperinflammatory stage in the body. And itolizumab is safe in COVID-19 patients. As I indicated earlier, the infusion reactions are manageable by slowing down the infusion rate. And in terms of summing it up, itolizumab effectively controls this hyperactivation of the immune system that we talked about in response to the COVID-19 virus, and prevents the morbidity and mortality that is related to cytokine storm. I think this is again a comparison in terms of how do we differentiate itolizumab and tocilizumab, but I would not go through this slide because Kiran has already gone through this part. Thank you very much.
Thank you, Sandeep, for such an insightful presentation into the clinical study. And it was great to know that all the patients who were on Arm A who received itolizumab actually recovered. And the only deaths that happened in Arm B were the ones who actually, unfortunate ones who did not get itolizumab. So that actually explains how excited we are with the results of this study, because we feel with itolizumab it will really enable us to save many lives. These are the patients who are actually progressing from moderate to serious, and if once they move to the serious stage of the disease, it gets very difficult to get them back. So thank you so much, Sandeep. And before we move on to our next session, let me introduce our panelists, where our principal investigators and other experts will share their experiences of actually treating patients with itolizumab. So for this session, we have with us Dr. Suresh Kumar. He is Medical Director of Lok Nayak Hospital, Delhi. He's a senior faculty member of one of the most prestigious medical colleges in the country, which is Maulana Azad Medical College, also known as MAMC. And he brings with him a wealth of experience in infectious disease. He also holds the unique distinction of being one of the premier COVID doctors of the country, as well as a COVID survivor himself. We also have with us today Dr. Rosemary D'Souza. Dr. Rosemary D'Souza is the head of the Medical Intensive Care Unit at Nayak Hospital in Mumbai. She brings with her over 22 years of experience in internal medicine. She has published numerous papers on the subject and is affiliated with Topiwala National Medical College. We also have with us Dr. Hemant Deshmukh. He is Dean KEM Hospital, Mumbai, a vascular and interventional radiologist and endovascular specialist. He is the author of multiple articles in peer-reviewed medical journals. We also have the privilege to have with us today Padma Shri Dr. Shashank Jyoti, who is the president of the Indian Academy of Diabetes and the Endocrine Society of India. He is member of Mumbai COVID-19 Task Force and is leading the efforts of managing the current health crisis in the city. He is a senior faculty member of Grant Medical College and has over 23 years of vast experience. Joining him is Dr. Hemant Thakkar, an eminent physician practicing general medicine. He's an intensivist and has experience using itolizumab in over 20 patients. He has more than 3 decades of experience, and he has been the principal investigator in over 35 studies and published several papers. This session will be moderated by Dr. Sandeep Atale. And over to you, Sandeep.
Thank you, Seema. Thank you, Seema. So I think we can start with the first question with our investigators. And I would ask Dr. Suresh Kumar, who is on the panel here, And I'm Dr. Kumar, if you are on, can you hear us well?
Yeah.
Okay, wonderful. So I think one of the things that we wanted to ask you is there is a confusion out there in terms of which patients should receive which drug, right? And for the benefit of everybody here, could you share your views on where does itolizumab play a role and which specific patient should be actually receiving it?
First of all, I must congratulate all Biocon team and particularly Kiran Madam. In the month of March, when this concept was coming about the use of this drug, and we discussed this in length with Madam, and I like this concept that yes, it can really work in this situation because we were losing large number of patients, and MEMC and Loknayak being the largest COVID facility in the country. We have 2,000 patients, and no drug was available even in the March and April end. So we thought that this concept is, we have a brilliant idea and we must move forward. And we got all this approval, and Madam said that, yes, you must be involved in this study. So I was encouraged, and because we also have the plasma facility and other things also. So we did this study, and after this, we found that this molecule is having great efficacy and safety. And the number of patients requiring this drug was much more than, because we have to follow the strict protocol. We were not using this drug on the ventilator patients. So your question was which group of patients we can use this, and where it have the maximum effect.
Yes.
So the patients of moderate severity, we enroll these patients, and we have the maximum benefit in those patients who are actually not having the cytokine storm. They don't have the organ damage or the multi-organ dysfunction. So the moderately sick patient, COVID patients, not on ventilator, were the group where we given this drug, and the results were very encouraging.
Yeah. No, thank you, Dr. Suresh Kumar. I think we have, you absolutely said the right thing. I mean, we really believe that this product, when it is given in patients who are about to enter a cytokine storm, and before the full-blown cytokine storm sets in, I think has the maximum benefit. So I think patients getting in a hospital who are on oxygen and who are presenting with with moderate to severe ARDS, these are the right patients from a timing point of view when they should receive it. So thank you for your answer.
That's
thank
correct.
you very much. Thank you. I would like to ask the next question to Dr. Rosemary. Dr. Rosemary, are you online? Can you hear me?
Yes.
Oh, great. So, Dr. Rosemary, you have been a really active doctor in Nair Hospital, and you've been treating so many patients in the last several months. And I think for the viewers, I think you have been an active trial investigator as well on the itolizumab trial. So can you share your experience of using itolizumab in patients in Nair Hospital?
Yes. So thank you, Sandeep.
Sure.
After getting the IEC approval for the trial, the first patient was enrolled on the 18th of May. We recruited 10 patients in total, out of which one was a screen failure because of a low lymphocyte count, absolute lymphocyte count. Out of these 9 patients, 6 of our patients entered the treatment arm with itolizumab, and the other 3 were in the control arm. Now, only patients who had a PF ratio of less than 200 were included. Most of these patients required ventilatory support in the form of BiPAP or CPAP, that is non-invasive ventilation, at the time of recruitment. The chest X-rays ranged from bilateral fluffy shadows to absolutely whitewashed lungs in some of the patients. Itolizumab was administered to these patients, that is the 6 patients, after confirming their eligibility. We needed to have these patients with a minimum white blood cell count to avoid complications of leukopenia. And after a week of infusion, in 4 of our patients, the clinical improvement was documented in the improving PF ratios. 2 patients, in fact, who were initially on BiPAP were maintaining saturation just on nasal oxygen at the end of the first week. And for the next 2 patients, they improved at the end of the second week. The daily SOFA score monitoring revealed an improvement from 3 to 4 for most people to 0 to 1 at the end of the second week. There was some fever with chills, which was a common side effect of the drug observed in the first infusion, but it was tackled well, and we were able to complete the infusion. This drug I found was a remarkable drug to save lives, and it definitely decreased the mortality, because we had 3 patients who were on the control, and out of those 3 patients, which we did not give the drug to, out of those 3 patients, 2 patients in fact succumbed to the disease. In those patients, the 6 patients who improved, even the X-ray findings were seen. In fact, at the end of the first week for 2 of our patients, and at the end of the third week for the remaining 2 patients. In fact, only one patient, the chest X-ray did not improve totally, but the patient had a past history of an ILD, so I wouldn't expect the chest X-ray to improve totally. And it was ensured that the patients were asymptomatic, maintaining saturations above 96% of oxygen, and the COVID test was negative on swab report before discharge. So it was observed that itolizumab definitely reduced the time to recovery and time to discharge for the intervention arm patients. A thorough phone call revealed that our patients who were discharged are also in good health. So our experience with this drug has been extremely encouraging, and I would recommend usage of it in all eligible patients. Thank you.
Thank you, Rosemary. Thanks for your nicely exhaustive answer. Very much appreciated. So I would like to ask if Dr. Hemant Deshmukh is there online as well. Dr. Deshmukh, are you there online? Looks like not, but we'll see if he comes back later on. But Rosemary, if you don't mind, I have one more follow-up question for you. You have enrolled almost about 9 or 10 patients on this trial, and even after the trial was concluded, you have kept using it in Nair Hospital. Yes. Can you just tell me what is the kind of response that you've seen in the further treatment of other patients beyond the trial itself?
Yeah, even we've been using it a lot. In fact, today itself, I gave one more patient the drug. The trick is that you should give it before the cytokine storm starts. Mm-hmm. When the patient is getting more and more breathless and hypoxic, I don't even wait for the result to come. I mean, we send the IL-6 levels just to document it. But it is given, and definitely the time to recover, the mortality is decreased. Forget the time to recovery. Even the mortality, I feel, is definitely decreased with this drug. So we've been using it quite often, even after the trial.
Okay. Now, thank you for that. Seema, do we have, since Dr. Deshmukh seems like he is not online right now.
Can I add one
yes.
or two? Yeah.
Yes.
So after this completion of this study, we were using this drug in 20 more patients, because many of the healthcare workers, nurses, and 3 of our doctors were having this COVID infection. So we gave this drug to doctors and our own employees, and we found it very effective. They were demanding that the results are so good that even our doctors, residents, and other staff requested for this drug, and we were purchasing from our hospital, and we have given to our own hospital staff. All patients recovered successfully, and there are only minor this allergy reaction in one patient, this chills and rigor in one patient, and that was also managed very successfully. So it's a promising molecule, novel biological, and it's very good results we have observed in last 1 month. So it's good to say that it's more and more physician and pulmonologists must use this. It's very good.
Thank you, Dr. Suresh.
Sandeep, if I can take a minute. So what Dr. Suresh Kumar said, we actually have a patient story, a patient who was treated as hospital. So during your session, whenever we can ask that to be played.
Hello.
After this.
Hello, Dr. Sandeep. This is Dr. Vishal. Can you
Yes.
hear me?
Yes, I can. Please go
Yes,
ahead.
yes. Yes, I am Dr. Vishal Gore working in Solapur. It is a bit periphery, not a metro city, where I'm using this itolizumab since last 20 to 25 days. I have used it in 23 patients. As Rosemary Madam said, we are not waiting for the results of IL-6 or inflammatory marker. At this periphery, when we are working in Solapur, we don't have laboratory facilities to check for IL-6 levels very early. We have to send it to Pune or Mumbai. We cannot wait to deteriorate the patient. So when we see the patient's oxygen requirement is suddenly rising, when he needs from 5 liters to 10 liters O2 to maintain the PaO2. So this is the right time to introduce the itolizumab, I think, because when we were trying itolizumab on ventilatory patient, for first two patients it didn't work that well. So one of them died, but one of them survived. So other 21 patients, we are using it when oxygen requirement suddenly goes up and patients other sepsis, bacterial sepsis and all parameters are ruled out. And at that time, we introduces this itolizumab and it is doing fantastic job. I would like to say that after 24 hours, the FiO2 requirement goes down by 20% in all these 21 patients. All are doing well. Many are discharged. Many are getting follow-up on phone that they are doing very well. 2 to 3 are still admitted in the hospital. They are on oxygen, nasal oxygen, only 2 to 3 liters. So all patients' hospital stay get reduced after using itolizumab. For those who didn't use that itolizumab, many got succumbed in initial stages. We were searching in Solapur. It was very difficult to get these drugs. So when one of my friend from Biocon, I called him. I said him, I want this drug because I have studied the pathway which it works when it was initially put forth. This can be used, and then we thought it is more promising for deteriorating patients, not already deteriorated.
Right.
So that part we clicked up, and we were using this drug since last 20-25 days, and for 23 patients it made magic.
That's very encouraging to hear, and thank you for your insights. I just want to expand this question to Dr. Thakkar also, because I think Dr. Thakkar has used in patients who are on a ventilatory support. And Dr. Thakkar, if you could just comment on that piece as well, because I think that would be important for our listeners.
I think at the outset, what I'd like to do is thank two people for initiating this innovative idea. Dr. Mazumdar has, of course, manufactured the drug and kept it on the shelf for psoriasis, but it was Dr. Joshi who conceptualized, and he approached me first, and then Dr. Mazumdar approached me. And they all felt that this would work as a monoclonal antibody because it works higher up in the cascade.
Mm-hmm.
So that's when we decided that we would take up this. Dr. Joshi and I discussed how to grow up. And I've used it in the trial for 21 patients. I've used it independently for another 5 patients. So we have about 25 plus patients. Of course, we lost where you had told me that. You just announced that. But where I would like to position this drug is around the 5th day or later. The bedside condition of the patient tells you that he is probably building up the cytokines in his system. His inflammatory markers are piling up. You don't need the IL-6 to be measured. You can see it clinically by the increase in respiratory rate, the increased oxygen demand, the tachycardia, maybe unrelenting fever. And that's when you could draw the blood again for the IL-6 marker and the other markers, but you immediately administer. I must tell you that amongst 25 patients, I've only had to use it a second dose after 7 days in one patient. Mm-hmm. So for me, ipso facto, it's a single dose monoclonal antibody which does the trick. And this is to prevent the patient from getting onto the ventilator. If you give it in time, then no matter how much the inflammatory markers generated by the body from the corona stimulation, no matter how much they bombard the receptors are blocked by itolizumab. So the patient doesn't tilt into obvious cytokine storm syndrome. You cage the virus, you tame the virus, and you trap the virus. During this time, the body gets over, starts building immunity, the virus burns itself out. If this started on the 6th day, you hold the patient's hand to the 11th day, and after holding his hand for 5 or 7 days, 10 days are over. You don't need another dose. The patient is out. For those patients who were already on the ventilator when we gave it, or we could not give it before getting them on the ventilator, the ease of removing them. Our 4th patient, by the time we got the shipment, he had already been put on the ventilator. And you know, ventilator is like a death sentence in the early part of our COVID battle. So in April and in May, I started my first patient on 12th of May. And we knew it was like a death sentence. So I had to intervene and I had to request Dr. Joshi to speed up the requirements. And he sent it to me. And within 12 hours of injection, 3 days later, we found improvement and the patient got off the ventilator. So I think it is preemptive. It is not for everybody. 85% plus of my patients will heal on their own. The remaining 12% will be admitted. They will be sharply and keenly watched with saturation and everything else. And only when they need it. The one other caution I want to exercise it, give the infusion slowly. Anybody who in a hurry decided to make Rome was not built in a day. You give it in 3-4 hours, it causes an issue. Because the patient gets fever, chills, tachycardia, tachypnea, and then you go from the frying pan into the fire. So in the later patient, we gave it over 6 hours and there was no problem. Like all other monoclonal antibodies, you have to give them, of course, a shot of steroid, antihistamine before giving it, because you don't want to burn another issue there. So when we gave it slowly, we had no issue. And I think this is a great drug which will form a pillar for treatment in cytokine storm prevention and in attenuation of the cytokine storm.
Right.
I used it for so many. I will continue to use it. And I'm thankful that Dr. Mazumdar and Biocon have brought this drug. And I'm also thankful to Dr. Joshi for setting up this entire trial and conceptualizing it. I'm sure we'll go a great long way with itolizumab.
Thank you, Dr. Thakkar. I think you beautifully explained your experience and you also simplified it so well. So appreciate it and thank you so much. I think, Seema, you have some patient experiences to be shown as well. So
Yes,
may I take your leave? I have other patients to go to. If I am done with, I would like to bow out, please.
Okay. Thank you, Dr. Thakkar. Appreciate you joining.
Thank you so much, Dr. Thakkar. Sandeep, in case you would want, I suggest you continue the discussion with Dr. Joshi.
Okay.
Then we can do that.
Great, great. So we'll do that. I want to introduce Dr. Shashank Joshi, who is working very closely with COVID for a number of months now. And he is an extremely active member of the Maharashtra COVID-19 task force as well. And we have some other participants who have joined here. Dr. Om Srivastava from Jaslok Hospital, he is here. We have also Dr. Vishal Gore from Solapur and Dr. Rahul Pandit from Fortis. And I think if they are here, Dr. Joshi, I would request that we spend about 15 minutes from now to really discuss, what is the armament of drugs that we have today, and actually what is the role of itolizumab in this whole spectrum of drugs that are available today. So if you could just engage our other speakers and get this discussion started, please.
So sure, thank you, Sandeep, and I again congratulate Kiran Madam and all of y'all for this novel idea. And I think we have been discussing over the last 4 months, every second day, how we can get better options for treatment. So let me come out with 4 disclaimers in the beginning. Number one, as we are even today on 13th of July 2020, we don't have a cure for COVID, but we have definitely novel options to save lives. And currently our endeavor is to ensure that we want to save lives and try to get our mortality down in India to less than 1%. So in our Save Life campaign, the viral drugs or the antiviral drugs, as Sandeep rightfully said, like favipiravir very early or remdesivir a little later, have data on the virus reduction. Remdesivir has some data on the recovery time, but both the drugs don't have data on saving lives or mortality. So that data is still forthcoming. Then we have the recovery trial from United Kingdom, which is only in the mechanically ventilated patients with dexamethasone. It can save lives, and most of us have been using methylprednisolone, and that has been a little earlier than that. But when the patient is desaturation, which Dr. Vishal Gore and Dr. Hemant Thakkar were saying, that's the right time to probably pick up a trigger for cytokine storm and probably administer drugs like itolizumab, and that's the space which also tocilizumab has been tried. Again, it doesn't have data, it has ongoing data on mortality. Our data and pivotal trial from Cuba and India is small, but it does show a positive direction. A lot of people might ask why such a small trial done, but in the current regulatory environment, as a repurposed drug for psoriasis from 2013, probably this was a regulatory requirement. But you can see off-label use by Dr. Gore from Solapur, Dr. Thakkar from Mumbai. They have definitely tried it out on a larger perspective. I have 2 of my task force members who are very well read in this subject. One is an infectious disease specialist, Om Srivastav, and one is a critical care specialist, Dr. Rahul Pandit. So Rahul, I'll start with you. Rahul, I want you to let our viewers know how do you use itolizumab in the treatment paradigm of this whole space of COVID-19, and have you got any personal experience off label with this drug? Dr. Rahul Pandit is the chief of critical care at the Fortis Hospital in Mulund, and it's one of the premier centers for treating COVID in Mumbai.
Good afternoon all, and first of all, my congratulations to Kiran Madam and the Biocon team for bringing out this drug and completing the trial. The data is fairly impressive as far as the proof of concept goes, and also that we have some early now data available for mortality benefit, which is being seen. Now, if I look at the treatment paradigm of COVID-19, there are 3 or 4 stages the patient goes through. So you have the first 3 or 4 days where the symptoms are mild, and you may have fever, or you may have some other constitutional symptoms like loss of taste, loss of smell, body ache, myalgia, whatever. Then there is usually a 3 or 4 days time where you probably are looking as if the patient is improving. And by the time you enter the second week, some of the patients, and we just don't know where this immunity switch happens, where the virus actually triggers the immune response in the body, and whether it happens in the first week or whether it happens towards the end of the first week, we just don't have any idea of it right now. But some of the patients tend to go into what we call as the cytokine activity or the cytokine storm syndrome. Now, it usually begins with the involvement of the lungs. Not all patients who have lung involvement go into cytokine storm. That's also true. So you have first instance is a drop in your oxygen saturation. And if you look at the treatment paradigm, that's where the steroids have worked the best. So if you have a patient who has the ventilator, who is either going to be on a ventilator or who has early hypoxia, that's where you may possibly consider to start them on steroids, because that's where the evidence is currently pointing out towards hypoxic patients. But the role of antivirals, the 2 antivirals, favipiravir and remdesivir, though there is no mortality benefit shown, the common belief and the common practice would be that for mild patients, you would use favipiravir in the first 5 days, and remdesivir probably from the 4th or 5th day for moderate to severe patients, definitely in a supervised hospitalized environment, not on an OPD basis. This is not a drug which you would be giving for outpatients. So having given steroids and remdesivir or antivirals, both are different category of drugs, you still don't have any control over the cytokine activity. The cytokine storm is probably still going to go. What we are hoping by giving antivirals is that the severity of cytokine storm may go down, which is what we are actually hoping for giving a remdesivir on time. And when the cytokine activity starts up, which starts up usually between 7th to 12th day, you have a new onset of fever, which is usually high grade fever. You may have worsening of hypoxia. You will have X-ray which starts going worse as well. And if you have done a CT scan, the CT scan becomes more and more involved. And along with that, you have some of your biochemical parameters like the IL-6, CRP, D-dimer, ferritin. They all start rising as well. Now, just the rise of IL-6 or CRP and a clinical well-being does not mean that a patient is a cytokine storm. So both of them have to go hand in hand. And once you identify that this patient has started to become either hypoxic, more hypoxic than what he was, or having a new onset fever, and say the CRP is risen up slightly, that is the time you need to start thinking about suppressing the cytokine storm. If you miss the boat at that time, you're basically going to have the storm come up. And then there is no point in giving whatever drugs you give. And then people have given one dose, second dose, and they still say that the patient remains hypoxic. So you've got to time it correctly. Time it when the storm is just ticking in. Don't allow it to sort of get established. So that's my take on itolizumab. About my experience, I've used it in around 6 patients with very, very pleasing results. As most of the other co-panelists mentioned, a very quick resolution of hypoxia and X-ray picture does happen. I have still got 2 patients who are still admitted with me. There is a concern that with every monoclonal antibody, you are going to suppress the immunity, whether you use tocilizumab or itolizumab, whatever. So some amount of care has to be taken to avoid secondary infection. You have to be extra vigilant to control this secondary infection if at all it happens. So those common things in intensive care have to be done, Dr. Doshi.
Thank you, Rahul. Dr. Om Srivastav is an infectious disease specialist, and Rahul started with a little concern that you also have not only interest in infectious diseases but also clinical immunology. So Om, how do you view that when you give agents like itolizumab or other agents, do you view it this secondary infection as a concern, and what is your personal take on this molecule and its novel mechanism of action, and how does it differentiate it from tocilizumab, which is available with us?
Okay, so thank you, Shashank, Dr. Joshi, for your introduction, kind words. Congratulations to Dr. Kiran and to the entire Biocon team on a molecule. I'd like to make a couple of points. Simply from the point of view as an immunologist, as a clinical immunologist, Dr. Joshi and we have been discussing this, number of times, that it's not just the interleukin-6 that we're talking about in the cytokine. There are multiple pathways that are expanded because of COVID and the inflammatory mechanisms that therein lie in that part. And the interleukin-6 antagonist, when you look at an antagonist like tocilizumab, it is a mono pathway inhibitor that inhibits only the interleukin-6. The advantage of itolizumab is that it functions at more than one, in fact, 3 pathways that we know about, and probably others that we don't know about. So I think that it is, insofar as what we are going to do in positioning, what the cytokine storm may be doing is something which is very important insofar as the pathways that are expanded by the inflammatory part of COVID. So in that respect, itolizumab, to my mind, is far superior to all the other agents that are available. For my own, personal results, I'm going to share an anecdote with all of you. A very prominent doctor that I was looking after in one of the South Mumbai hospitals actually started collapsing while he was talking. He was talking and then he collapsed. He had to be shifted to ICU and ventilated immediately. It was the day where Mumbai was asked to be preparing for the cyclone that was coming this way. And I think that Dr. Praveen will recollect this patient, because I did ask him to urge the distributor not to shut down, because I needed the itolizumab pretty much right then. And the distributor was kind enough, after the request of the Biocon Periphery team, and we managed to get that molecule, the itolizumab, within about 2 or 3 hours. We gave it to this doctor immediately in ICU. He made a fantastic recovery in the next about 7 or 8 or 9 days. And at that time, after he recovered, we were having a chuckle that, look, if we can manage to get the itolizumab in the middle of the cyclone, it's almost certain that you're going to make a full recovery. So good, very, very personal, very encouraging results that I have seen. Itolizumab is very important insofar as understanding how to prevent those parts. So we tend to play a lot of attention to the cytokines that will affect the lungs. We probably need to look, and I'm going to urge Dr. Joshi and Dr. Kiran to probably see if we can start an arm of even an observational study of other organs which are affected. My own feeling is that the biggest advantage of itolizumab is going to be in predicting a vascular path. People who are going to get cerebrovascular accidents, people are going to get myocardial infarctions, pulmonary embolisms, deep vein thrombosis, mesenteric artery thrombosis. The entire thrombotic process of COVID is probably so inherent, so integral to the cytokine storm. And I have a feeling that itolizumab may be best poised for preventing this part, because while we are concentrating on the lungs, and that's probably the biggest organ to be involved, we probably need to pay a little more attention on the vasculopathy that comes with COVID. And I think that itolizumab has a great role in preempting and stopping those complications from happening. So I think that if Dr. Joshi agrees and Dr. Kiran agree, we should probably look at structuring this. Having said that, my own experience with itolizumab has been very gainful. It has been very encouraging. I think there is a definite role, especially because we tend to disregard the immune part a little bit. We probably need to focus a little more on that part. Itolizumab, of course, like any other monoclonal antibody that suppresses the T cell responses, it is important. It is imperative that you cover with an antibiotic, a broad spectrum antibiotic, before you give the molecule, because a good about 2/3 of the patients will go into septicemia, or at least be prone to septicemia, if you're not careful about that part. So covering for a secondary bacterial infection is critical. Other than that, I wish the molecule all the very best. I think there is a definite place for itolizumab in treatment, not just of COVID. It'll probably have a place in a number of infectious diseases. And once again, my heartiest congratulations to both Biocon, Dr. Kiran. Thank you, Dr. Shashank Joshi.
Thank you. So we have with us also Dr. Vishal Gore. Vishal, my question, you very nicely told us where you can use this molecule. Now, Vishal, can you tell us that you are in Solapur, where cost matters, and you know that government also is now giving tocilizumab, which is very expensive. So compared to tocilizumab, definitely itolizumab is much cheaper. Each vial is around 7,500 rupees. We need around 4 to 5 vials, so the cost works out to be around 25,000-30,000 rupees. So it's definitely much more cost effective. Remember, biologics are more expensive than the rest of the therapies which we have. But what are your experiences? You have used them off-label in 20 cases, and today, as I said, our mission now is to save lives. Our mission in India, across India in COVID, is to try to get our mortality below 1%, and that's really the larger mission. And we have these molecules which are Make in India, which can be life-saving. So would you like to share some more data, Dr. Gore, from Solapur?
Yes, as I said, I'm working in Solapur, and the cost of therapy matters here a lot, because many people are from low socioeconomic strata. But the illness itself is killing everyone. So initially, when we have 110 patients in our institute right now of COVID, initially what happened, people were dying in front of us. People were deteriorating. People were not affording tocilizumab 40K drug, which may need second dose within the 12 hours. And at that time, we were losing people. So at that time, I just called my friend who is one of the working from Biocon.
So Vishal, how expensive is tocilizumab now? And compared to, say, tocilizumab?
Yes, tocilizumab, one dose in Solapur, it is around 45,000. People are getting at 45,000 for one dose. For two doses. Yeah, so many times you need two. Yes, 90,000 for just a drug, which may or may not work. To convince the people that this drug may or may not work, you have to spend 90,000. People are not ready to pay.
And now it is in short supply also.
Yes, now it is in short supply. For your kind information, one of my patients got it in some other market in 1 lakh rupees for one injection. This is terrible. So we were frustrated initially because of all these things. Then we came across this itolizumab. I have studied the pathway which it works. Then I thought it is working better than the tocilizumab, where we are getting patients who are worsening on the day 2, day 3, because they are coming to us on day 8, day 9. So at that time, when we started using itolizumab, thanks to Dr. Praveen, he make it available in the Solapur on the day we require it. Initially, people from Solapur were running to Mumbai and Pune, because no drug is available here in Solapur, like itolizumab, tocilizumab.
Hello.
Yeah.
But after started using this drug, I can proudly say people who are doctors or who are from all class of the society are staying here in our hospital because of seeing the results of our bad patients in last 20-25 days. We are retaining our patient here. We are treating them very well. If you listen to all panelists, everyone was saying that the right time of the drug is most important part. Use it. When there is increased requirement of oxygen, then when there is requirement of BiPAP, suddenly this is the right time. You need not to check the IL-6 levels and all these things. Clinical worsening with correlation is the most important part. Many times we are getting surge of the cytokine parameters next day. But if you use it on the day when oxygen requirement goes up, patient needs BiPAP. At that time, when we use itolizumab, it is helping. And the cost-wise, if as Muzumdar madam said, it is 7,800 for a 25 mg. Here in Solapur, we are getting at 7,500 rupees for one vial. So for 3 to 4 vial, it is as per the weight of the patient. Many are like 40, 50 kg. So for them, we are using 75 mg, and more than 50 kg, we are using 100 mg. No patient required second dose after 7th day. For our patients, when we used for 23 patients, no patient required second dose. All were doing better. As I said, only one got died, who was on ventilator, who took AMA discharge and went to civil hospital. There he died. I don't know the further things about him, but all other 22 patients.
So congratulations to you, and thank you very much. You must write up your paper, and it is wonderful.
Yes, sir. Yes,
One
sir.
last
Yes.
question to Dr. Rahul Pandit. Rahul, if you are there, this 6-minute, it is not possible in places like Solapur to do interleukin 6 levels and various biomarkers. So can we use this 6-minute walk test which you have popularized across Maharashtra? And can you tell us to our national viewers something about this 6-minute walk test and desaturation? And can it be used as one of the indicators to use early use of tocilizumab like medications? So can you just tell our viewers how to identify that deteriorating patient clinically through a saturation 6-minute walk test and biochemical? It's a very relevant question in COVID. It may not be therapy specific, but that probably is the right time to use it, because the most important thing here is the right time, as Om said, as Vishal said, and I think Professor Suresh also said the same thing. Suresh Kumar, Rosemary, they all are saying the same thing. Hemant said the same thing, and I don't know whether Dr. Hemant Deshmukh has joined us before we can open it for media questions. I'm sure there are a lot of media questions, but Rahul, would you like to just share with us?
Absolutely. So the 6-minute walk test is nothing but it's basically giving a stimulus to your cardiorespiratory system. It has been used for ages together. In fact, the American Thoracic Society actually puts it up as one of the tests to assess the cardiac as well as the pulmonary function in patients who have chronic heart disease or chronic lung disease. Now, we have extrapolated this test into COVID era. Now, what have we done is that we are actually asking patients to walk for 6 minutes at a steady pace, at their own steady pace, within the confines of the area in which they are, because obviously they are isolated and they are quarantined. So they are usually walking up in their own room. The test is done by recording a saturation before the start of the test. And the saturation should be at least above 94%. Then the patient should not be on oxygen, and patient should be able to walk properly unassisted. Walks for 6 minutes. At the end of 6 minutes, you look at the saturation again. If there is an absolute drop of 3% or more from the baseline, or if the saturation goes below 94%, it is called as unmasking the hypoxia. That means the cardiorespiratory reserves were not enough to maintain the body's needs of oxygen. The body is getting starved of oxygen because the lungs are basically not able to take in oxygen too much. So that is a time when you think of these patients requiring higher care. Now that higher care could be in form of either admitting them to the hospital if they are in the OPD basis or at home, or if they are in the hospital, you need to think of these therapies, whether you're going to add steroids, whether this is the time where the cytokine storm is kicking in. So I would not use 6-minute walk test to actually give tocilizumab or itolizumab, but I would certainly use that as a trigger that yes, this patient is probably heading for a cytokine activity, because now he's having hypoxia, which up until now was unmasked. Now you have unmasked it, and it was a mask, and now you have unmasked it, and you have actually shown to the people that yes, this patient is getting hypoxic. So let's use that as a marker to identify the patient. Most of the times, our patients will be in ICU or in the ward, will not be able to do a 6-minute walk test to actually determine whether hypoxia is there or not for a monoclonal antibody to be given.
So maybe a CRP. You'll have a cut point for
so
CRP.
What needs to happen is that along with that, we need to do CRP, and if the CRP is going up, oxygen requirement is there, you can very well send the IL-6 level at that time, but that would be the time to time your monoclonal antibody, itolizumab. That is the time when you need to time it up. If you time it later on, then it's a problem. And if there are seniors above 60 years of age, then you can basically shorten the 6-minute to 3-minute walk test.
So we are running on Indian stretchable time, 22 minutes late as
Dr.
per our
Joshi,
communication.
I think, yes,
so
I was just about to say.
We need to wind up. So we don't. You just. I think Dr. Hemant Deshmukh has joined us. So if you want to ask a question, and you can open it up to the. I think you and Kiran can take the questions probably.
I think we should do that, because I think we have a number of people who wanted to ask questions. So we'll open it to the general question session now. And Seema, do you want to moderate this session, please?
Yeah, first of all, thank you very much. That was very, very insightful. All the doctors really brought in a lot of, analysis and their experience of dealing with. So without further ado, let's start with the questions. Sukrita, would you moderate that, please? Just prioritize the questions. A lot of questions have already been answered during the course of these discussions. So let's go to the questions which have not been answered. And due to shortage of time, if there are some questions that we're not able to answer, I can promise all the journalists will come back to you. You can drop in an email and we'll definitely get back to you.
Yes, absolutely. As Seema said, we will answer all the questions, and we will also be organizing multiple scientific webinars after this one and share the invite details with you, where you will have the opportunity to ask more questions. So without further delay, the first question comes from Samreen Ahmed of the Business Standard. And Samreen asked that Remdesivir is being sold at sky-high prices in the black market. What steps will Biocon take to control the price of itolizumab in the market? What kind of demand do we anticipate? And what is the production capacity? Kiran, would you be able to pick up this question for us?
So as you know, obviously we are going to make sure that this is going to be very patient specific. As you know, we need to have patient consent. We have to have the prescriber signing off that they need the product. So it will be done through a very closed channel. And we will ensure that, there is no question of people trying to black market it. And if we find any such activity, we will take strong action. Secondly, we are ramping up capacity as much as possible, because as you know, we hadn't, expected this to see the compelling results so soon. So we do have capacity right now to address the present, size of the serious population. But we do realize that this is a much bigger challenge, not just in India, but for the world. So we are going to ramp up capacity as much as possible and as fast as possible to make sure that we can make it available for India first, and of course globally.
Thank you, Kiran. And the next question that we have today is from Avik Das of Times of India. And he asks, very related to how you close the last question, is which countries will this drug be used in? Are we seeking approval outside of India and Cuba?
So right now, Avik, we are focusing on the Indian, needs. And of course, we will, I'm sure, get requests from other countries. As you know, our US partners are also planning to basically carry out a similar kind of a larger or a US-based clinical study, a clinical trial. So based on that, of course, if the US approval comes, there will be a huge demand. So we are at the moment focused on India. If we do get inquiries from other parts of the world, we will be looking at that.
Thank you, Kiran. And the next question that we've got is from Sumi of the New Indian Express newspaper. And Sandeep, I would request you to take this up with the panel. She asks, many health experts have said the itolizumab trial with 20 patients in the intervention area is statistically too small to draw any scientific conclusions. And the approval by the DCGI seems rushed without phase 3 trials. What would you have to say about this?
Before Sandeep responds with his statistical explanation, let me please first make it very clear that this is an emergency situation, and in an emergency, the size of the trials are always small, because that's why it's given restricted emergency approval. I also want to mention here very emphatically that the regulatory process is extremely robust. The kind of scientific discussions that were had on this trial were of a very high order. I now hand it over to Sandeep.
Thank you, Kiran, for doing that. So I can say that, firstly, I think I would like to point out that this is a label extension of an already existing product. So we have a lot of safety information over the past seven years on this product. The second part is, as Kiran mentioned, I mean, there are instances where there is, whether it's an orphan disease or whether there is an emergency use, there are other statistical models that can be used. So when we approached this trial, we used statistical models to show that if we are looking at mortality reduction. So if we can show a magnitude of effect in such a small trial, which is statistically significant, then even a small sample size would suffice. Now, had we not seen that magnitude of effect, which we saw in this trial, then obviously we would have to expand this trial to have more patients in it. But I think all said and done, when we look at these results at the end of this trial, what we have clearly demonstrated for the primary endpoint as well as some of these secondary endpoints is we have shown statistically significant differences and advantage for itolizumab. And therefore, I think when we had these discussions with the regulators as well, they were convinced that the magnitude of effect is really high, and therefore the approval came through with this smaller trial.
Thank you, Sandeep.
Mm-hmm.
We have a question from Tabassum Barnagarwala of the Indian Express, and she asks that the price of the drug is not affordable for poor patients. How do we plan to provide this drug for this class of patients? Kiran, would I direct this question to you, please?
Yes. So I think the first thing that people asking these questions must also ask the question, how much does it cost the government or the private sector or the private paying patient the cost of staying in an ICU setting for a number of days? I think nobody takes that into question. Now, you heard every doctor saying that you can actually discharge these patients much sooner, and you also are saving lives. So remember, if you can rescue them from going on to the ventilator, the cost of putting people on a ventilator is also very high. I'm sure the other doctors can actually add to what I'm saying. So in relation to that, a cost of 25 to 30,000 rupees to save a life and to reduce the number of days spent in an ICU is hugely beneficial and not expensive. Maybe Shashank would like to add to this.
So I think three things. Number one is that cost of ventilator care, hospital care clearly gets reduced, and saving life is the paramount importance. So I think from that standpoint, compared to say a tocilizumab, which is 3 times the cost of itolizumab, clearly this is a cost-effective method to say the least. Secondly, I think the government of Maharashtra has got it for the Bombay Municipal Corporation as well as government. Even tocilizumab and remdesivir has been bought by the government and made it free available to all our public hospitals. And Tabassum, you are aware of that. In fact, they have stocks for a month already available with them. So that's something which we know. So I think the government is ready to spend. They know that investing in some of these life-saving molecules actually will reduce the healthcare burden on the overall healthcare infrastructure. So from a cost economics perspective, it is definitely there. And coming to the other question of the small sample size, I think in a pivotal trial, when we want to answer a question, the minimum statistical number required is 30. and therefore a lot of people might still critique that 20 and 10 is too small a number. Yes, I agree with that, but as long as you apply a robust statistical design where the phase 2 and phase 3 has already been tried out in 2013 on more than 500 patients, I think it gives us a proof of concept that whether it is working or not. And the VALT test, which was applied by the group, clearly shows, because I was on the data safety monitoring board, is what is the first research question we asked: is it safe? And the answer to that was yes, it is safe. Is it effective? The answer to that is yes. Thirdly, does it reduce morbidity and hospital recovery time? The answer to that is yes. Fourth, does it save life? Yes. So this small trial has given us insights into that for us now to use it in a labeled expansion. Now, a lot of other doctors you saw have used it off-label because they had nothing else to do to save their lives. For example, the doctor from Solapur, Dr. Hemant Thakkar, and many others, they've used them off-label in more than 20 cases, and they've clearly seen that they have been able to save lives. So when it is, or you can see Dr. Rahul Pandit or Dr. Om Srivastav, many doctors have used them off-label because currently our endeavor is to save lives of those severely critically ill patients with cytokine storm, because we don't have too many options available with us.
And I just would like to add that if you add all these patients who have benefited from the drug, it is over 150 patients. So we will be collecting all this data to add to the current trial.
Thank you, Kiran, and thank you, Dr.
Regarding
Shashank. That was
the cost of the drug, that not all patients requiring this drug. Those who need ICU care or ventilatory care or going to be in the ICU. So if this drug reduces the stay by 2 or 3 days also, the cost of drug is already incurred. Yes. So I don't think the cost of itolizumab for poor starter, it is the problem. So itolizumab working better, reducing the hospital stay, saving the money of the patient. I think this is more important.
Thank you, Dr. Gore. That was a very important statement.
Yes, thank you, Dr. Gore and Dr. Shashank, and thank you, Kiran. I think that was very beautifully explained. We now take the next question from Seema Singh, who asks that our data is pretty good, and what more do we require in terms of study length, sample size, or anything else to take this from emergency use to standard of care? And her second follow-up to that is that given that we would like to sell this in other countries, will the Indian RCT data suffice, or what more is required? Sandeep, may I direct this to you?
Sure. So first of all, I think with the approval of this product, this has been approved as an emergency use approval right now in COVID-19. So what that means is this product can be used as long as COVID-19 is there, and it is for the emergency use. We also are committed to do a phase 4 study that came as a commitment to the approval. So we will be starting a phase 4 study as well. In addition to that, our partners in the US, they are committed to starting another global study where they would be doing multiple countries as part of that trial. So we will be generating more data. Obviously, as Kiran alluded earlier, when there are inquiries from outside, from other countries, we will have to look at what would be needed in terms of additional data that may be needed. But at this time, I think we are committed to serving our obligations with a phase 4 trial, which will gather more data. And with our partners, we'll be initiating a global trial as well. So I think that being said, there will be more data generated, which can be utilized for other countries.
And I would also like to add, Seema, that many of these doctors who are on this panel are going to be publishing their work, because I think it's very, very important that we get this scientific temper and this research temper to global attention. And I'm really happy that many of our doctors here, whether it's Dr. Vishal Gore, Dr. Hemant Thakkar, Dr. Rahul Pandit, many people, and Dr. Om Srivastav, everyone has actually done some very interesting studies on their own. And I think that data needs to be published. So I think the more you publish the data, I think the more robust will be the acceptance of the data.
Thank you, Kiran. That's about all the time we have for the Q&A today. Like we said, we will answer all of your questions via email, and we will have scientific webinars to follow where you will get the chance to ask your questions again. At this point, I would like to hand it back to Seema, who would like to showcase a patient video and deliver the closing address. Thank you.
Thank you, Sukrita. Let's now hear a story of hope, one of the patients who was actually recovered after being treated on itolizumab.
नमस्ते, मेरा नाम है। मैं 15 जून को पॉजिटिव आया कोरोना का। एक हफ्ते मैं घर में ही रहा और घर में ही रह करके मैं इलाज करता रहा, लेकिन कोई फायदा नहीं हुआ। मेरी तबीयत बहुत ज़्यादा बिगड़ गई थी, यहाँ तक कि मरने की नौबत आ गई थी। मेरा बेटा मुझे लेकर आया हॉस्पिटल और यहाँ पर मेरे को एडमिट कर दिया। यहाँ पर एक नई दवाई से मेरा इलाज किया गया, जिससे कि मैं आज बिल्कुल ठीक हूँ और अपने घर जा रहा हूँ बच्चों से मिलने के लिए। मैं यहाँ के सभी डॉक्टरों से बहुत बहुत धन्यवाद करता हूँ, सभी का, कि इन्होंने मेरी बहुत जी जान से सेवा करी।
Thank you, Sukrita. I hope most of the questions have been satisfactorily answered by the panelists. And now I would like to invite Dr. Christiane Hamischer, CEO and MD Biocon Biologics, to share her concluding remarks and put the conference to an end. Over to you, Christiana.
Thank you, Seema. So first of all, on behalf of Biocon, I would like to thank all our colleagues in the media and also in particular the expert physicians for sharing their experience, but even more for their continued partnerships in the past week and months. And I'd like to thank everyone who joined us in the audience today. I very much hope that you agree with all of us that what we have heard today from our expert physicians is giving us ample hope that we are at the beginning of a very, very impactful journey that absolutely has the potential to save many lives in India and also across the globe. And it is the need of the hour to repurpose medicines like itolizumab. Itolizumab is a breakthrough first-in-class novel biologic therapy that got the restricted emergency approval for the treatment of patients first in the world with moderate to severe COVID complications. We still see this alarming number of COVID-related deaths, and we are looking very much forward to the continued partnership and the collaborations with the experts to benefit the patients in India and globally. All of us at Biocon and at Biocon Biologics, we are aware of this huge responsibility, and we are glad and we are also proud to be part of the work that so many teams have done so far to bring us to this first so important milestone for itolizumab. And during a time when it matters the most, and the COVID crisis is one of these times, at Biocon, we are and are always committed to enable affordable access to the innovation that is needed. And at the moment, this is itolizumab. Thank you so much for joining us today. And with that, we are closing our press conference.
Thank you. Thank you, Christiane.
Thank you.